Our first speaker in this session is Joshua Jarek from Indiana University. And he's going to talk to us about G protein coupled receptors and particularly directed evolution as a tool to explore these receptors and their conformational dynamics. And so Josh, I'll let you go ahead.
Okay. Are we looking okay?
Yes, we see you.
Okay, great. Hopefully you see my screen too. Okay, first off, I'd like to thank the organizers for giving me an opportunity to talk about my research. I was really hoping and looking forward to the conference in Nashville. I really loved it last year, and you really can't beat being able to bring the whole lab to such a good conference so affordably. But I guess if you think about it, it's even cheaper, free registration, and keeping everybody in Bloomington. So this worked out pretty well, I guess, financially. So okay, so today I'm going to be talking about some of our research looking at GPCRs using NMR. The title may be a bit misleading because I really don't go into the advantages of directed evolution so much, but hopefully you'll glean some of those out of the talk as we go through it. And so before I really get into things, I just want to introduce the people who have been working on it. So this is my lab. We're just starting our third year at IU, so it's a bit rough around the edges. The work I'm going to be talking about today is mainly work by my postdoc Fabian Bumbach and my PhD student Austin Dixon, and both of them received a lot of help from another postdoc, Scott Robson, in the collection and processing and analysis of the NMR data. So, okay, a little bit of GPCR biology first. I like to think of GPCRs as molecular watchtowers. So they're membrane proteins. They exist on the extracellular, the plasma membrane, excuse me, of eukaryotic cells. And structurally, they're comprised of 7 transmembrane helices, okay? And so they're watchtowers in that orthosteric ligands, or extracellular ligands, will bind to this orthosteric binding pocket here. I hear that my mouse isn't great, so I'll wiggle it profusely, so hopefully you'll see where I'm pointing. But so these ligands that bind, they're really diverse in their properties. They can be small molecules, they can be peptides, they can even be photons of light. And when they bind to this orthosteric pocket, they cause some sort of conformational rearrangement of this transmembrane region and, allow intracellular proteins to bind on this surface, okay? And these intracellular proteins are known as transducers, either G proteins or arrestin. So this is where the receptors get their name from. And when those transducer proteins bind to the activated receptor, they cause some sort of biological response, which really depends on the ligand that is recognizing them. So why do we care about GPCRs? I like to tell the NIH that I care about GPCRs because they dominate the biochemical classes of drug targets for therapies that are currently on the market. I guess I say currently, but this looks like this paper is about 20 years old. But you can easily see that GPCRs make up at least a quarter of the drug targets for therapies on the market. And what the table below really highlights is that these the drug targets are not restricted to a particular disease. You really see that they're ubiquitous. So we see autoimmune diseases, neurological diseases, cardiovascular diseases. So these proteins are really everywhere. The other reason that we care about GPCRs is they're just really cool. They're exemplary allosteric proteins. Merriam-Webster has this beautiful definition of allostery being something that is relating to, undergoing, or being a change in the shape and activity of a protein that results from combination with another substance at a point other than the chemically active site. Okay, so other than the orthosteric site. So when I was a biochemistry graduate student taking biochemistry classes, I was exposed to 2 forms or mechanisms of allostery, like an induced fit model of allostery. The idea being, if you have 2 conformations, an inactive and an active conformation, in the absence of a ligand, the two states are so thermodynamically separated that only the inactive state is populated, and that you require the active state in order to you require the ligand in order to populate the active state. And then the other model of allostery that I learned was conformational allostery. And the idea here is that the thermodynamic barrier between the inactive and active states is much less. And so it's possible that both of these states can exist at a standard temperature. And then the ligand can come in and bind, and it can stabilize the active conformation. And so this idea of conformational selection or allostery shouldn't really be that crazy to this audience. I think spectroscopists are trained to think in terms of ensembles. If we go back with the GPCR story, if we go back 30, 40 years before there was any sort of structural insight, there was pharmacological hints of allostery within these proteins. And so you have a classic two-state biological response where in the absence of a ligand, you would have no biological response. And as you titrate in the ligand, you would saturate some biological response. You get a nice, beautiful sigmoid like this. And what the pharmacologists saw long ago was even in the absence of a ligand with a GPCR, you would still get some basal activity, okay? GPCRs have very rich pharmacology where it's not just simply full agonists modulating this basal activity, but there's competitive agonists or neutral antagonists that just sit at the orthosteric site. They can turn off this biological activity with inverse agonists. And then of course there's molecules that bind at the orthosteric pocket and do some sort of intermediate activation of the biological response. In addition to this, more recently, the pharmacology has gotten even richer in that there are allosteric modulators on the extracellular surface, and actually they can bind at regions even in the transmembrane that will further modulate the signaling or the biological response of a ligand. And so this is just to kind of orient you with this figure here. This dashed line indicates the agonist at a given concentration. So for example, the EC50, okay? And then there are positive allosteric modulators that can then increase that signaling. There are negative allosteric modulators that can decrease that signaling. And there's even, again, silent allosteric modulators that don't seem to affect that particular biological response. So incredibly rich pharmacology hinting at allostery in the GPCR family. But then moving forward in time, once cryo-EM and structural biology, X-ray crystallography caught up with the pharmacologists, they really underscored this idea of functional allostery, which wasn't much of a surprise. If you solve the structure of the receptor in the inactive state, it looks like this. If you bind a ligand at the orthosteric pocket, you see some subtle structural rearrangements. The main one is this transmembrane helix 6 moves outward away from the transmembrane bundle as compared to the apostate. And then if you add in a transducer protein, you see an even more substantial shift of this transmembrane helix 6, though for the most part, changes in the rest of the protein aren't really apparent in these average structures generated by cryo or X-ray. And the other thing I just want to highlight here is these light gray proteins I illustrate with each of these structures. These are stabilizing molecules. So none of these structures could be determined in the absence of T4 lysozyme or nanobodies that are locking the receptor into a given conformation. And that in itself really underscores this ensemble view of the receptor. Okay, and so then, if we're progressing in my timeline of education, when I actually got into a lab in graduate school, I learned about dynamically driven allostery. So this is something that didn't make it into my biochemistry textbook. But the idea here again is this is kind of similar to conformational selection in that the thermodynamic barrier between the inactive and active states is low enough that they could be populated to some level. But the key point here is that the width of a well in these free energy landscapes reflects the conformational entropy. And so if there's a lot of conformational entropy, that effectively lowers the kinetic barrier between these two states, allowing these fast timescale motions to also modulate these conformational changes. Again, this might not be the slide for this audience. I don't think I need to convince you of the awesome power of NMR, that not only can we get information about the thermodynamics of given states, but we can also get information about the kinetics of the interconversion between these states. And so this is a really nice technique to couple with static and molecular dynamic simulations. So here's a really brief summary of applications of NMR, solution NMR to GPCR signaling, by no means exhaustive. One thing that I want to highlight here is that these molecules are eukaryotic molecules and can really only be expressed well in insect cells, to some level in yeast cells, but unfortunately that really limits their labeling potential by NMR. So that for the most part, people have been using some particular amino acids that they can add in during the expression, or probably more commonly is some sort of chemically reactive groups, either like reductive methylation or these trifluoral groups that they can add to a cysteine. And the results that they've seen can kind of be summarized a little bit here. This is a study by Stefan Grezic's group, and this shows that the chemical shift perturbations can actually track very nicely with efficacy, at least in this case. That's why I have this little asterisk here. So there's other studies that show that the chemical shift does not track at all with the efficacy of the ligand. Here's studies from Kurt Vuschik's group using fluorine NMR, and this is showing that in the absence of a ligand here with this particular receptor, there's two states in slow exchange, and that the populations of these states in slow exchange can be modulated by the additions of ligands. And then here's kind of the opposite by Scott Proser's group, where they looked at a different receptor, in this case the beta-2 adrenergic receptor, again looking at fluorine labeling. And here, rather than seeing slow exchange like what Kurt saw, he saw something more, or they saw something more intermediate. And so when they did CPMG studies and they simulated the spectra and calculated the delta omegas, they ended up seeing multiple states that were in this, this intermediate, fast intermediate timescale. And so for our work that we're going to be talking about today, my interests are in the neurotensin receptor. It's primarily expressed in the gut and in the brain. It's a peptide binding GPCR. And so naturally, the binding of the peptide stimulates hyperglycemia and hypertension and vasodilation. And it has some involvement in various neurological disorders. One of the main things that works out very nicely for us is that this is one of the few receptors, I believe 3, where there's structural information for both transducer complexes. So both the G protein bound structure and the arrestin bound structure have been determined by Cryo-EM, as well as structures of the ligand bound receptors. So this is really a great place for us to start. But unfortunately, like I kind of mentioned a couple slides ago, these receptors can only be expressed in eukaryotic systems, and even then they're incredibly unstable. And so what we decided to work with is this evolved construct of the neurotensin receptor. And the first thing that this gives us is this improves the thermostability quite substantially. So this evolved construct I call EN-NTS1 as composed to just NTS1. Okay, you see a 30-degree shift in thermostability, but yet it's still able to bind to ligands and still able to promote signaling responses. So it's not a bad place for us to start. And so my story today is going to be focused on this neurotensin receptor, this evolved neurotensin receptor, and using NMR spectroscopy to look at thermodynamic and kinetic changes at 3 different locations along the receptor. So the extracellular, this orthosteric pocket, the transmembrane region, as well as the intracellular side. I'd love to give you some beautiful stories of how we've figured it all out. Unfortunately, a little bit of a spoiler alert, we haven't. So this is where we'll start. So we'll start with the extracellular region. So this is the labeled neurotensin peptide. So this is very advantageous for us that it's a peptide binding receptor, so we can look at the ligand or use the ligand to look at the orthosteric pocket. So here's an HSQC of the ligand. What we do see is we're able to visualize all of the amides, which is great. The chemical shift dispersion is obviously unstructured. We see the cis-trans isomerization of the proline. If we take it then and we add in the receptor, the first thing we see is most of the peaks disappear. So we're left with just these 2 peaks in blue. I put question marks next to them because Fabian is actively working to assign them right now. So unfortunately, we don't know who they are, but what we do see is that they have very substantial chemical shift perturbations from any of the nearest neighboring resonances. But one cool thing that did pop up when we added in the receptor is the side chains. So we have these 2 arginines, arginine 8 and arginine 9, and we're able to see the epsilons of both of them when we add in the receptor, which obviously highlights that there must be some sort of hydrogen bonding or salt bridging that's going on that's reducing the exchange kinetics and allowing us to visualize these resonances. Fabian then went a bit further and he threw in arrestin, the transducer, to see what happens. And so looking at the backbone amides, we've now reduced ourselves to a single amide, again, of unknown origin. But we do see chemical shift perturbations of both that backbone as well as the side chains. And we also see some suggestion of changing to the line shape of these. We don't know whether or not this is a change in exchange or dynamics, or whether or not this is a change in just sheer molecular weight of the system. So this complex probably tumbles someplace around 80 or so nanoseconds is the tau C on this. But this is really cool for us, because if we just look at the structure that's been determined, the nearest atom from arrestin, or the closest arrestin gets to either the backbone amide or the side chain arginines, is at least 40 angstroms away, and we're seeing these beautiful allosteric effects. Now, obviously, this isn't necessarily unexpected, right? So Gurevich, more than 20 years ago, showed that if he takes the ligand and competes it off the receptor, shown in these open circles here, okay, that if he takes that receptor ligand system and he adds in arrestin, he'll actually shift the affinity for the ligand 1 to 2 orders of magnitude. So we already know that this allosteric communication from the ligand binding pocket to the transducer binding site is reciprocal, in that binding of the transducer will cause a change in ligand affinity. And also we see already hints of this from the structures. So there are distinct conformations, average conformations in these structures between the ligand receptor in yellow and the ligand receptor, the ternary complexes in blue or in purple. And especially in these arginine side chains, we see large shifts. There's no density for arginine 8, but we see a large translation of arginine 9. And we see that all the way down into the core of what would be the transmembrane region, in both the isoleucines and the leucine here. We see that the rotameric angles of these are different. But our interests are more along the dynamic side, right? And so this is data that Fabian is actively collecting again. So in addition to trying to figure out the assignments right now, he's trying to kind of fill in this bit of information. So these are just some curves from the side chains. So these are T1 and T2 measurements oh, I realized I put these in the wrong place. The dotted lines are the ligand with the receptor. The solid lines is the ternary complex, okay? And so what we do see is we see changes in the T2 values. We need to reproduce the experiments for the T1s and for the tracks. So I don't want to go into any overinterpretation of this data here, but just showing that working with this peptide and only seeing these two resonances is actually quite advantageous in this case, where we can collect 1D spectra and we can directly measure, for example, from the tracked experiments, the cross-correlated relaxation rates. And so Fabian's collecting this data right now, and we'll see where we go with it. And so then the other project that Fabian's been kind of working on is looking at this dynamic allostery from the orthosteric pocket to the transducer pocket to the intracellular pocket. Okay, and MD simulations have really suggested that there's a role for dynamic allostery, but it hasn't been determined for GPCR systems yet. And so he's doing this using methyl labeled methionine. And so here's the neurotensin receptor. It has 10 endogenous methionine residues. Here's the HMQC spectra. You see there's not 10 peaks, so there's quite a bit of overlap. So he applied a strategy of removal of the methionines and then reintroduction of them into the system. And so this is just showing how he went through and assigned each one of these methionines. And so in the end, we settled on a system as kind of illustrated here with 7 of the endogenous methionines. 3 of them were removed because of peak overlap. And then, what he did is he collected all of these assignment spectra with the various ligands. So this is quite a blur to look at here. So we took some artistic license and made a nice little illustration of it to kind of give you some direction. And so, again, unfortunately, this is all very new data, and I hesitate to overinterpret it, but I guess I'll just point out a couple features in that, we have some residues such as M244, which is relatively close to the ligand binding pocket, and that seems to reflect nicely or correlate nicely with the ligand efficacy. We see this nice linear line between antagonist and agonist, but then we also see residues which are actually also close to the ligand binding pocket. So 330 is right up here in the ligand binding pocket. And you see that there is no correlation between ligand efficacy and these chemical shift perturbations. So we're currently going through analysis to come up with some plots and figure out some correlations here, but we don't have a good story to tell. But what we're trying to do in the meantime with now all of these assignments is we're actually trying to collect some dynamics information. So we're ready to start some forbidden states experiments to get some cross-correlated relaxation rates, put some order parameters on this, and then in collaboration with Nagarajan Vaidehi, she's been collecting some MD simulations for us, and that's what's shown here. So the methionines are highlighted in pink. You can see the ligand here with the one of the side chain arginines really swinging around quite a bit, but making contacts with various loop regions up here. And then this is a little plot just to kind of validate our MD simulations in that we've collected some experimental functional thermostability data, and it correlates very nicely with the internal energy of these systems. So we think that kind of validates our MD simulations a little bit. And so in the last couple moments, I'll show you the third side, looking at changes in dynamics and conformation in the intracellular half. And so this is done with fluorine labels here. And so here's where they would be in the various states. And our question is, can we measure these large conformational changes at TM6, where it kicks out when it's bound to the G protein or the arrestin? Can we measure these? Hopefully my graduate student isn't too mad that I have distilled his heroic 6 or 9 month effort of getting the labeling right into a single slide here. But this is just to show that he can label just the single TM6 site. So here's the probe that he has added in. He introduced a cysteine here, did a mutated a glutamine to a cysteine, and there's where the label is on TM6. And so then he started collecting some fluorine data. So this is the unadulterated fluorine 1Ds. He takes this information and he uses Meister-Nova to deconvolute it into its composite resonances, something like this. And as he does this, he basically fits an increasing number of states until the residuals, shown here in red, are pretty equal to the noise within that spectrum. And then the program also simulates a spectrum, which is shown in gray here. And you can see that there's really nice correlation between the simulated spectrum and the raw unadulterated spectrum. And then to kind of validate the results of the states that we pull out of this, what we try to do is we take a single peak and we fix its chemical shift value. And then we allow MaestroNova to refit where it can fit the chemical shift, the line width, and the intensity of the other two peaks. And basically we sit here and kind of move the chemical shift of a given peak to confirm that we're at the right location. And so we get these nice little wells as long as our signal to noise is high enough. And so here's what some of that data looks like.
2 minutes.
Thank you. And so here's what some of that data looks like for various ligands here. So here's the populations of each one of these states, and then here's the line width of each one of these peaks. And so again, this is, I hate to keep on saying this, but this is all brand new data, and I really don't want to go through over-interpreting this. To be honest, when we look at it, we just kind of scratch our heads a little bit when we look at it. So we see this one state pretty much dominates regardless of what the ligand is. And we see these little perturbations in this S1 state, this downfield shift and this upfield shift. But for the most part, we don't see major changes, and it's difficult to draw trends. The line width seems to suggest a correlation perhaps between ligand efficacy, but we're still trying to work on interpreting this data. But Austin did go ahead and he threw in some transducer molecules that bind it to the intracellular site, and we do see some pretty substantial changes that kind of are just, they're very interesting. We see some large population changes where when we add in the transducer, we see the state 3 really grows, suggesting that it's the active state. But at this point in time, we have a collaboration with Oliver Ernst in Toronto to do some DREPR to try to get some structural information of the ensemble that we're looking at in solution. We also have some collaborations to figure out, to do some cellular assays and figure out kind of functionally what these states are. And so the other thing that Austin's kind of working on at the same time is just dynamics. He's been collecting T1s and STDs to try to look at slow timescale interconversion. You see that there's really nice plots here. We just haven't fit any KEX to them. And so just to kind of conclude, our goal is to combine NMR and MD in order to look at and elucidate the allosteric mechanisms of GPCR signaling. And we're looking at it, we're hitting it from all sides, extracellular, transmembrane region, the intracellular half. But we obviously have a long way to go, and hopefully we'll be able to meet in person at a future conference, have a beer around a poster, and hopefully we'll have some interpretable data to share. So again, this is my team, and I'd like to take any questions.
Okay, thank you. I see one question just popped up. The question is, are your conformation diagrams showing allostery? Won't a new funnel coming up only upon ligand binding give a more accurate representation of allostery? In the absence of a ligand, protein doesn't exist in the other conformation. Or will they be having equal populations, such as feudal conformations? So can you comment on whether your data is showing allostery?
Yeah, well, I think the data is definitely showing, if our very basic definition of allostery is an ensemble view, for example, then looking at these singles, this fluorine is labeled at the single site, And as we look at it, we see that there are three distinct states that are being populated. And thermodynamically, they're different. And they seem to have different kinetics. And so yeah, we see evidence of allostery by that definition. We just don't know any sort of mechanism for it at this point in time. I don't know if that answers the question or not.
Okay, and I have a second question from Chuck Sanders. Chuck, do you want to go ahead and ask a question?
Sure. Thanks, Josh. That was a really nice talk. I know that's really hard getting these systems set up, and it's nice to see it set up. I sort of have just a question about your intro. So, and that has to do with the unavailability of E. coli as a way of labeling GPCRs. So, I know Andreas Plukton in Zurich expresses highly engineered GPCRs in E. coli, and there's even like a crystal structure of one of these. And I thought that like Gerhard was collaborating with him at some point to try to do NMR on these using uniform labeling. I was wondering if you know what the story is on that work and why has it not gone forward?
Yeah, well, you're totally right. So I was working with Gerhard, or when I was doing my postdoc with Gerhard, we were doing that collaboration. So it's the same receptor. This version that we're working on here is actually slightly different than what Andreas has been using. And so, with that work, basically what it came down to is that receptor is functionally inactive. The version that Andreas and Gerhard were working on. And that's not to say that obviously it doesn't yield any interesting insight into these systems, but it kind of slowed down because of that. So both myself and the other postdoc who were working on the project started our own independent labs, and that work kind of slid down a little bit. But Franz has published recently on it, actually in what is it in? It's not in Angewandte. Franz Hagen published recently with Andreas Plukton, though, just in the last month or so. But the journal for that paper is escaping me a little bit.
Okay, well, thanks very much. I really appreciate getting an update on that.
Yeah, thanks, Chuck.
Okay, I had one last kind of quick question, philosophical question. So it might be a naive question, but so you're interested in the molecular dynamics of these GPCR proteins at different spots, and you mentioned on either side, and then in the transmembrane domain. And so the way I'm sort of viewing this is that the dynamics or changes in molecular dynamics might be important in terms of ligand recognition and binding on either side or inside of the membrane or the outside of the membrane. But it would seem to me that the membrane itself would damp transmission of dynamics changes across the membrane, so that they might not be as important in terms of actually influencing the signaling, transmembrane signaling, so much as the interactions on either the outside or the inside of the membrane. Can you comment on that?
I guess the one comment I could make, if I understand, I guess could I ask a question? What you mean as, or what you believe is in terms of the membrane dampening the?
I'm wondering. Yeah, sorry. Are you proposing then a mechanism by which a change in dynamics of either on the outside of the membrane or on the inside surface of the membrane is somehow being transmitted across the membrane?
Yeah, I mean that's what the the MD simulations kind of suggest that a little bit, is that this is that they do see basically a correlated change in side chain dynamics that goes through the transmembrane region of the receptor. So what they're trying to illustrate, at least here, is, you know, if they look for correlations of dynamics through that transmembrane region, so on the left of this figure, in the absence of a transducer molecule, they see multiple different pathways that seem to have very subtle correlations to them. But then when they add in transducer molecules, like what's shown on the right here, what they see is that a lot of the subtle pathways kind of lose their coherence, and that there seems to be one or two very strong pathways of correlated dynamics that go from the orthosteric pocket to the transducer pocket, to the intracellular side of the receptor. So,
okay. So cool. So that would be a very long range transmission then.
Yeah.
Yeah, that's very interesting. Okay, well, in the interest of time, we better move on to our next speaker. So thank you very much, Joshua.
Thank you.
The second speaker in this session is Kathleen Hall, who's for many years been at WashU in St. Louis, and she is an expert on experimental molecular dynamics, particularly in RNA. And so she's going to talk to us today about her work with backbone dynamics in an RNA recognition motif, and how they facilitate protein binding and RNA recognition specificity. And so, Kathleen, the floor is yours.
Okay, so thank you so much for your invitation and your patience here. Okay, and the people that did the work on this protein dynamics is basically Greg DeCoster, who's the NMR expert. And you'll see he's done a yeoman's share of assignments and dynamics measurements. And this is a collaboration between us and crystallographers who are in Germany, Gerd Weber, Nicola Holton, and Marcus Wall. And they've made like 5 crystal structures, co-crystals, and we are going to use them in some of our interpretations. So why do we need to understand RNA binding proteins? And these are some of the reasons, because as people find more and more RNAs, and it turns out all those RNAs are somehow now associated with proteins. So that means there is a real push now to try to understand how those proteins are working, how they're doing their recognition, where they're doing the recognition. And the problem is that there are very, very few examples of the mechanism of RNA recognition by proteins. Very, very few examples. So what I'm going to tell you about is the work that we've been doing on a particular protein. These are our RRM proteins, RNA recognition motifs. These are folded domains, and they're often present in proteins that have more than one. And often in proteins that have other kinds of domains, and often those are intrinsically disordered domains. But what I'm going to talk about now is just one of them. And what's important about these proteins and what makes them distinguishable among all the other alpha beta sandwiches out there is the fact that they have aromatic residues that are exposed on the surface of the beta sheet. And these amino acid residues are actually stacked with the nucleic acid bases. That's one of the ways we can distinguish them and recognize them. So most of these RRMs recognize short single-stranded RNAs, and the affinity is often weak, and they are typically very promiscuous in what they bind. And I'm giving you a couple of examples here if you're familiar with these. But the one I'm going to talk about today is unique in many ways. It's called SNF, which is an RNA binding protein. It uses a single RRM, although it has 2. A single one is going to bind RNA. And the way it's going to do that is really one of the reasons we were very interested in this is because it's one of the few that we know about, maybe one of the only ones we know about, that binds with very high specificity and high affinity to two different RNAs. And we wanted to understand how it did that. So the RNAs that it binds are shown here. One is from the U1 SNRP and one is from the U2 SNRP. And you can see that these are hairpins. The part of the RNAs that, the part of the RNA that gets bound is really the hairpin part. And the two hairpins differ in their loop closing bases, in the C to G substitution, and also in the insertion of an A. And so these are highly conserved, phylogenetically conserved in these particles. And the recognition is key to formation of the SNRP. So in many higher metazoans, there are two proteins, one that's unique for the U1 and one that's unique for the U2. But in lower metazoans, it's only one protein. So what do we know about SNF RRM1? And our SNF here comes from Drosophila. This is again a representation of it. What we know is that its folding free energy is fairly low, minus 3.5 kilocalories per mole, and when we do an unfolding experiment. And we have to do their unfolding here in urea because guanidine basically just denatures the poor thing. If we look at its amide HSQC, it's really well dispersed. So this is a fairly well behaved protein domain in some ways. And that some ways is a caveat and is shown here. So the protein itself, again, like I said, it has two RRMs. They don't interact with each other. They're completely independent. RRM2 does not bind RNA. RRM1 does. And if you look at the intensities and the breadth of the amides, you can see there's a pretty horrendous difference between the two. RRM2, very sharp peaks, very intense peaks. RRM1, very low intensity and very broad. And so looking at this spectrum, you might suspect that something is going on here with exchange. And what you see when you look at the backbone by delta R2s is that, yes, in fact, that whole backbone is pretty flexible. So here we're measuring the delta R2 effectives for the proteins for the protein RRM1 at two different field strengths, 700 and 900. And you can see that throughout the protein, there is a lot of motion going on at this kind of time scale. And particularly in alpha 1, which is sort of strange because it should be packed up against the beta sheet. And also here is one of the loops, which is probably not too surprising. But again, throughout the body of the protein, there looks like there's a lot of motion going on a fairly slow time scale. And again, looking at alpha 1, we're going to get back to why that's important, but we can actually use CPMG relaxation dispersion to measure the exchange. And what you see here in two of the residues in that alpha helix is an amide exchange on the order of about 1,000 per second for these two, just fitting them to a two-state model. But that was strange because when we looked at other RRMs by the same kind of method, we don't see any difference. Well, we don't see this kind of very localized motion dynamics. So if we look at the crystal structure, on the other hand, the crystal structure of RRM1 has 6 proteins in the unit cell, and only loop 3 shows any kind of conformational heterogeneity. So we've got a protein that we know that now there's conformational heterogeneity in loop 3, and the whole thing is sort of floppy. is one way of putting it. Okay, so what do we know about how it binds? Well, this is a crystal structure of the RRM bound to the U1 stem loop 2. And what you can see is that loop 3 is tucked up inside the RNA, which is not too surprising. There are a couple co-crystal structures of the U1A protein, and we see the same thing. Here is the C-terminal tail, and here's alpha 1. I don't know if you can see can you see my cursor? I don't know if you can.
Yes, we can.
Yeah, we can see it.
Okay. So here's alpha 1. And so alpha 1 is, you can see, no, it just disappeared. So here's the one end of it, and it's pretty close to the RNA. What happened was, when we tried to make a crystal structure of SNF bound to stem loop 4, we couldn't get it to stabilize. We could never isolate a complex for crystallographic studies. And that was frustrating, but maybe not too surprising, because when we measure the affinity, we always find that stem loop 4 binds weaker than stem loop 2. This is nitrocellulose filter binding, and it's in fairly high salt, 250 millimolar KCl, because if you drop the salt concentration too low, then the binding for stem loop 2 is too tight to measure. So we always see a difference. In this case, it's about 50-fold weaker binding for stem loop 2, stem loop 4, but that's significant because in the nucleus, the biology is such that in the nucleus, there's an overabundance of stem loop 2 as part of the U1 SNRP. The U1 SNRP is probably at least 1 million copies per cell, whereas the U2 SNRP, where the U2 stem loop 4 comes from, is at least 10-fold less than that. So if you have a great abundance of the U1 and a weaker binding of the stem loop 4, the stem loop 4 might not even get populated in the context of the nucleus. And so that's a biological problem. So if we look in the NMR now, because we can form the complexes in the NMR, if we look at what we see for the binding of stem loop 4 and stem loop 2, what we find is that in terms of the chemical shift perturbations, they're pretty similar. Here's the deltas for binding of stem loop 2. Here it is for stem loop 4. Here's loop 3, which is basically what we'd expect. It's sitting here in the middle of the RNA, and it's making contact with the nucleic acids bases and the backbone. So here's stem loop 3. Here is the tail, the C-terminal tail. And here is beta 1. Now both of stem loop 2 and stem loop 4, pretty much the same. They're sitting in the same place on the protein. There's a little bit of difference, and in terms of who's interacting with what. Most of the differences are in loop 3, and as we're going to see, that's pretty much what we'd expect. The dynamics of the protein when the RNA is bound, the fast dynamics as measured by heteronuclear NOEs, they really only change in 2 different places. One is in loop 3, and the other is in the C-terminal tail. Binding of the RNAs locks those fast dynamics down, so they're really damped, which is again not too surprising because we know those are going to make contacts with the RNA. What we see with the slow backbone dynamics is something quite different. There's a very selective change in the delta R2s, And what we end up with when the stem loop 2 or stem loop 4 is bound is that most of those dynamics that we saw throughout the body are now almost completely damped. And those are mapped onto the structures here. But the only method, the significant dynamics that we retain are in alpha 1, which is now on the back, what we call the back side of the protein. The RNA, remember, is sitting on the front side, and the back side, there's no RNA there. So that was sort of peculiar, and it would suggest that even when the RNA is bound, that alpha helix may not be tightly packed in the interface. That interface may not be very well defined. So that's significant because, well, do these data suggest a rationale for how the binding affinities of those two RNAs are different? Well, I would have to say not really, because the binding sites appear overlapping via the chemical shift perturbations, and the protein dynamics are pretty similar. So getting back to alpha 1, it's a little odd, at least to me. I've looked at a lot of these RRMs, and they all sort of look the same. And that's another part of the problem in trying to figure out how they work, because their structures are almost superimposable of the at least 10 of them that are now known. And so the dynamics of alpha 1 are weird. So back to the biology here, in the U1 SNRP, SNF RRM1 is bound to stem loop 2. But in the U2 SNRP, the biology tells us that RRM1 is bound to stem loop 4, the RNA, but it's also bound to another protein. And that other protein is the U2A prime protein. So what does that have to do with anything? Does that have anything to do with binding? And it turns out it does. So this is the binding assay again of RRM1 binding to RNA, and this is stem loop 2. And now what we've done is add in the U2A prime protein. And you can see that there's basically no difference between having none and having 1 micromolar of auxiliary protein. It doesn't really seem to change the affinity of SNF for the RNA. That's very different for stem loop 4, because now what we're doing is titrating in U2A prime, and we're seeing a dramatic increase in the affinity of the protein for this second second RNA loop. So how is that working? Well, it turns out this is a crystal structure from the Vol lab of the LRR domain of U2A prime binding to SNF RRM1. And what you can see is that 2 things. One is that the structure of the RRM, whether it's isolated or bound to the LRR domain, is basically the same. You're not seeing any large conformational shifts that would explain a different binding affinity, for example. But what you are seeing is that alpha 1 is basically like a hot dog in a bun, as my students like to say, wrapped up by A prime, U2A prime. If we look at the dynamics now of SNF RRM1, when the protein is bound, what we see, this is SNF alone, and this is SNF now bound by U2A prime. The dynamics now are very different. And in fact, this was a very difficult experiment for Greg to do, because we did not deuterate these proteins. We didn't deuterate the LRR domain. It turns out it's difficult to work with. It tends to be It tends to aggregate, it tends to precipitate. And so we tend to keep it in fairly high salt. And you can see these experiments were done in 200 millimolar KCl. All the dynamics that we saw in alpha-1 are basically gone. Probably because we're now exchanging our protons with the bound protein. But we're also seeing now diminution in the dynamics of everybody else, except for alpha-3, which is the C-terminal tail. So the residues here, again, they're probably in exchange, probably due to conformational exchange. So I said that we've lost the dynamics, but I think what we've really lost is the intensity, and because they've gone into exchange. So we've sort of mapped those onto, again, the structure of the domain, and we remember this is the surface that the RNA binds to. So if we look at now the crystal structure of the two complexes, what we see is, here's our A prime, which does not make any contact with the RNA, by the way, and it doesn't bind to RNA. Here's our RRM, and you can see that there's heterogeneity in the crystal structures for where that C-terminal domain is hanging out. Here are the two RNAs that are bound. And there is a difference in the structure of the loops. And also in the position of, but not in the position, but in the interactions. And we'll see that a little bit later, of loop 3. So what we would propose, just looking at these structures and looking at our NMR data, is that when RNA binds to the RRM, it leaves alpha-1 flexible. Here's alpha-1, because it needs to be flexible to associate with the U2A prime protein. The flip side is that when U2A prime binds, it doesn't change the dynamics of the RNA binding surface, but it allows it to still remain flexible so that it can bind to the RNA. So we've got sort of a compensatory interaction going on here. And again, what's really interesting is that the structure of the RRM is not changing. It really is, it's not a question of large structural changes that are driving the interactions. And if we look at the interactions that we see in the co-crystal, which we can't see by NMR, what we see is that if we look at stem loop 2, we see interactions. Here's the stacking interactions of the phenylalanines with the bases in both of the different RNAs. What we see in this side of stem loop 4 is there are now a lot of interactions that are coming from the side chains of lysines, and then this alanine. And what we think is happening is because the alpha-1 is now bound by A prime, it's held in such a way that it can make interactions with this side of the loop. And in the absence of holding that down, the protein isn't able to actually make these kinds of interactions. And so it basically doesn't even see them when it's bound to stem loop 4. So what we conclude, again, by looking, this is just two structures of the RRM, now mapping onto them the dynamics when they're bound to stem loop 4 versus the LRR domain of U2A prime. There's basically an inversion of where the dynamics are maintained. So what we would suggest is that the ensemble of RRM1, the ensemble of conformations, is controlled by its binding partner. And the result is that there's cooperativity, and we would say this is allostery, that modulates the binding affinity and specificity, because this is really a case of conformational capture in terms of allostery. And so, again, this is not at all what we expected to find. And it's also, I think, the first example of a protein-protein interaction that is modulating an RNA recognition. And what I'm afraid of is that all these people who are looking for RNA-protein interactions are looking for RNA and protein and trying to understand how they're working. And I'm concerned that they're sort of leaving out other binding partners, and that what we're seeing with this kind of system is a modulation of both affinity and selectivity by other kinds of molecules that are there in the cell. And so I want to also mention that we've been working on this system for quite a while. We have contributions from Dr. Kim Delaney, Michael Rao, Tom Stump, Dr. Chang Tang, who's been running our NMRs, and Sandra Williams did a lot of the original binding experiments. Our RNAs for crystallization, and there were a lot of them, come from Agilent Labs, and this was funding by to Marcus and to me. And I'll just leave you with this picture of from the crystal structure. And I know I said these are structures that are really hard to get by NMR, and these little green dots here are waters. And what we found in the crystal structures is that there are waters everywhere in this interface. So trying to get interactions by noses, for example, by NOEs between RNA and protein is very difficult because a lot of the magnetization just goes into the water. So we've really relied on the crystal structures to help us figure out how this is working. But we've relied on the NMR to show us basically what I think is the mechanism of conformational selection and how that thing is working. So this is something that only NMR can give. So I obviously am very fond of it. So thank you for your attention, and I'm sorry this got a little bit confused at the beginning. So, are you there?
Thank you, Kathleen. And yes, let me apologize for the glitches at the beginning. We never know what happens in some of these Zoom presentations, and I think you showed great grace under pressure.
Yeah, sure.
Everybody's worst nightmare is
oh yeah, everybody's worst
the
nightmare.
technology won't work. So don't worry about it a bit. I believe we have a question from the audience from Gajendra Singh.
Okay.
Gajendra, you want to go ahead and ask a question?
Yeah, I can't see you, so I'm blind here.
I'm not hearing anything.
I'm not hearing anything.
No, is it possible, Gajendra, you could type your question into the question and answer tab, and then I can read it to Kathleen. And while we're waiting for that, Kathleen, I had a question myself, actually.
Mm-hmm.
So very interesting talk. So you mentioned the enthalpy of binding of these things. There's not a lot of, if I understood correctly, there's not a lot of enthalpy. So I'm wondering where the free energy driving this comes from. Are these
oh, actually, they're
entropy-driven processes? And then
no.
And then I'm wondering whether you don't think so. Then I'm wondering
Well-
whether it's coupled with the thermodynamics is actually coupled with the protein-protein interaction, which is helping drive the overall thermodynamics.
We've measured binding by a couple of different ways. One is by ITC, so we know the enthalpies. They tend to be large. There's also, there's a huge coupling constant related to these. The thermodynamics is very complicated because there's the protein, the residues on the surface of the protein are coupled to each other. This is something that Jim Krantz in my lab showed a number of years ago. There's a network of interactions on the surface of the protein that have to get, they can be disrupted. And so the binding actually depends on the temperature, whether or not you're enthalpy or entropy driven. There's entropy enthalpy compensation. The delta G's tend to be significant. The protein-protein interaction is mostly entropic, you're right. And that probably has to do with letting go of the waters in the interface. But the RNA protein interactions are often enthalpy driven, probably because of the stacking interactions. And there's an enormous hydrogen bonding network that drives these things. So there's no simple answer to the thermodynamics. Yeah, I know too much about it to say it's that simple.
Okay.
I'm sure that's not an adequate answer.
Well, I think it is a perfectly good answer. Okay, so I see we are still waiting on Gajendra's question. I'm sort of thinking at this point we should probably move on to the final presentation and let Gajendra talk to you privately with a question after.
Okay, that's fine.
So thank you very much. And let's
My pleasure.
let's see if we get Michael queued up. Go ahead with Michael's talk. Michael Kennedy is also an established investigator. He's going to talk to us about human rotavirus, a very important and clinical problem, and the domain recognition of the histoblood group antigen and host cell receptors by NMR. And and Michael's, again, is from Miami University of Ohio. And let's see if we can get the screen sharing going this time. Okay, we see your screen.
Okay, that's a good start.
And we can hear you. So
There we go.
the floor is yours.
Okay, good. Well, thank you, Mike and Don and Marcus, for the invitation to participate today. And I'm sure that everyone who has participated will join me in thanking each of you for doing an excellent job organizing this online conference. So what I'm going to talk about is the work that we've done in my lab over the last 5 or 6 years. I would say focusing on understanding the molecular basis for human rotavirus receptor domain recognition, and these happen to be histoblood group antigens. So just to begin with some significance, rotaviruses are the principal cause of severe diarrhea in children. And as recently as 2011, were responsible for approximately 200,000 deaths in children younger than 5 years old, primarily in developing countries in Africa and in India. In around 2006, 2008, 2 vaccines were introduced that reduced the burden of death. In 2016, it was reported that the vaccine saved around 29,000 child deaths, but also reported that a universally effective vaccine could have prevented an additional 83,000 deaths. So focusing on the rotavirus itself, it contains 11 segments of double-stranded RNA that encode 12 viral proteins, including 6 non-structural proteins and 6 structural proteins. Now, this structural protein VP4 is going to be the one that we're most interested in, and it's depicted here. And so this VP4 spike protein is the protein responsible for host cell recognition. So if we spend just a little bit more time on that, focusing on VP4. So this VP4 spike protein requires protease activation. And the result of that protease activation is exposure of the VP8 star domain. Here I have my VP4 protein here. So that protease action exposes this receptor binding domain, VP8 star, and then also VP5 star, which is a membrane permeabilization protein. So depicted here is the structure of the receptor binding domain of the VP8 star domain. And this is a common galectin fold that binds carbohydrates. And I just want to draw your attention to the figure on the right. In the red here, I'm going to be speaking about receptor binding domains that either occur at this beta-beta sheet edge, or at this area between this beta sheet edge and this alpha helix. So we'll be referring to a beta-beta binding site or a beta-alpha binding site. So another thing that we need to know about is that rotaviruses are classified as either G-type or P-type based on their antibody production. And so the rotaviruses that infect humans are the group A rotavirus strains. These are one of 10 rotavirus species. And so the G-type rotaviruses, that is the classification that responds to the VP7 protein, which is a glycoprotein, which forms the part of the outer icosahedral capsid. And then the P-type rotaviruses depend on the P4 the P4 protein, which is of interest for this discussion. So VP7 G-type and VP4 P-type. So I referred to 2 existing vaccines. So the first one that I'm going to talk about is the G-type vaccine. And this vaccine is based solely on the antibody response to exposure to the G-type antigen. Now, the other existing vaccine is Rotatech, and it's pentavalent, meaning that it has 4 G-type antigens and just 1 P-type antigen. So I'm going to refer to these as we go along. And so there are limitations of these 2 existing rotavirus vaccines. The limitation being that these 2 vaccines are reported to be highly effective in North America and in Europe, but they're also deemed to be largely ineffective in these developing countries in Africa and India. And the question is, why is that the case? And so to get a clue about this, our collaborator, Jason Jiang at Cincinnati Children's Hospital Medical Center, did a phylogenetic analysis of all of the known rotavirus genotypes. And the result of that phylogenetic analysis allowed him to group the rotavirus genotypes into 5 genogroups. And so group P1, genogroup P1 here, primarily infects animals. And then we have P2, P3, P4, and P5. P2, the P2 genogroup, has 4 genotypes in it, and these 4 are the ones that are primarily responsible for infecting humans. We have P8, which is the most common one and responsible for the most deaths, followed by P4, and then we have P6 and P19. This phylogenetic tree analysis led our collaborator to infer that this is an evolutionary offshoot from this most ancient P1 genogroup that emerged under the selective pressure to these histoblood group antigens. And so I should emphasize that this phylogenetic tree analysis is based on the sequences of these receptor binding domain sequences. So what might explain the limitation of these existing rotavirus vaccines? And our hypothesis is that the existing vaccines are limited because only one P-type genotype is included in the only one of the existing vaccines, despite the fact that there are 4 different human RV genotypes within that P2 genogroup. And because they potentially have distinct receptor binding specificities and potentially different antigenicities, that could explain the lack of universal protection by the existing vaccine. So the goal of the project was then to generate data to test this hypothesis. And our strategy was to determine the structures of the 4 different VP8 star receptor binding domains in complex with their host receptor molecules, and to determine the relative binding affinities to the various receptor glycans. So getting into the experimental section, then I need to introduce you to the glycans that we studied in this research. And I'm going to start over here. There are several different classes of histoblood group antigens. They're organized into H-type, A-type, B-type, and type 1, type 2, etc. The group that's relevant to the group A rotaviruses that infect humans are of the class these H-type 1 variety. And so ones I'm going to talk about in this study are shown here. And really the important thing to recognize is that if we focus on LNT here, well, there are a couple of things. The way these histoblood group antigens are generated is they start with a disaccharide precursor, and that's shown in the box. So all of these share this disaccharide type 1 precursor. But this one lacks what we refer to as a secretor fucose, which is shown in this green triangle. So some of these, they lack the secretor phenotype. And then the other one that's important is the Lewis phenotype, and that's this kind of pink colored, also a fucose. And so you'll notice some of these glycans have both a secretor and a Lewis phenotype, and some of them have neither. So we're gonna have secretor negative or Lewis negative, or secretor positive and Lewis negative, or secretor positive and Lewis positive. So that's the important thing to take away from this collection of structures here. So on to our experimental data. The first thing that we wanted to do was to study this P19 genotype, which was thought to be the earliest ancestor, evolving from the P1 genogroup. And what we're showing here is saturation transfer difference NMR results, binding to, in the lower case here, binding to the mucin core 2, and then in the uppercase, binding to this sequence which has the H-type 1 disaccharide plus the secretor fucose. And so these results show that we were able to detect binding in both cases, and we were able to map out the epitopes for binding onto the specific protons of each of the glycan structures. So that's what's shown in here in yellow and orange and so on. So we established binding, and then these galectin folds produce beautiful HSQC spectra, and we were able to do titration experiments and to do the complete residue assignments. And so we were able to establish the region where these glycans bind on the surface of this galectin fold. And also we were able to determine the affinities or the dissociation constants as well. So what we find here is the location of binding is what we referred to as that beta alpha binding site. So in this case, if we compared the location of the binding site to what was already known in the literature, the other existing structures were for where the sialic acid glycan, the sialic acid group of the terminal sialic acid group of histoblood group antigens, or A-type histoblood group antigens, they bound in a different location in the beta-beta binding site. But our data from the NMR experiments indicated that these glycans bind in a new site referred to as this beta alpha site. So since that was a new site, we needed to confirm that, and we did that based on site-directed mutagenesis. And so here, for the mucin core 2, which is shown over here, we can show that if we mutate the residues that were previously found to be involved in binding sialic acid or A-type histoblood group antigens, that there was no loss of binding. But here, if we knock out the residues we identified in our binding site, then we lose all binding. And then that was for the mucin core, the mucin 2. And then the one that was secretor positive but Lewis negative, we found the same thing, that we don't lose binding if we knock out or if we mutate the residues involved in sialic acid or A-type histoblood group antigen binding, but we do lose binding if we mutate the ones we identified in our study. So we were fortunate enough to also be able to solve the crystal structures of both the mucin core 2 structure and the secretor positive structure, and those crystal structures are shown here. And what we see primarily is that in the dashed box here, we see that the primary part of the glycan, which is common to both of them, that is responsible for binding, is this section, the disaccharide H-type 1 precursor. And so that seems to form the main interactions with the binding site in both cases. And so this just kind of summarizes showing our crystal structure data and our NMR-based mapping data, and showing that here, again, this genotype binds the glycan in a different location than either, in this case, this was P14, which was of genogroup P3, or P3, which was genogroup 1, or here we have P11, which was genogroup 4. So the genogroup 4 is over here in the beta-beta site. Genogroup 1 and 3 binds down here, and then this representative from genogroup 2 binds in a new location. So just one more point about P19 is that here we have an overlay of the binding of the mucin core in red in the secretor positive glycan shown in blue. And you can see the molecules hang off the binding site a little bit differently in either direction, but what's common again is that disaccharide H1 type precursor where it binds in that binding site. So kind of the big target for us was trying to identify the structure of the P8 genotype bound to its glycan receptors. Up until this work, which we just published in 2020 here, there was no structured data available for binding of the P8 genotype to its glycan. And so again, based on the saliva binding assays done by our collaborator at Cincinnati Children's, he identified the lead molecules, and we, in this case, we have a tetrasaccharide, which has both the secretor fucose and the Lewis fucose, and we have another one that's a little bit longer that also contains the secretor fucose and the Lewis fucose. And we did our saturation transfer difference NMR experiments and we confirmed binding, and we were able to identify the epitopes for binding with the red circles indicating those that experience the strongest saturation transfer. So we went on to do the chemical shift perturbation mapping on the surface of the protein. Again, we were able to assign all of the data and do the mapping onto the surface of the proteins. And what we see here is that it appears that both the Lewis tetrasaccharide and this one with both the secretor and the Lewis fucose, they now appear to bind in the beta-beta binding site, but there was some additional chemical shift perturbations away from the beta-beta edge, which was over in the region of the beta-alpha binding site. So just to make sure, we did a number of mutations. So we mutated a few different residues here that were over in that ambiguous area, like K168 here you can see, and T78 and D79. And what we found is that even in the presence of those mutations, we still saw the chemical shift perturbations in the same location. We still retained binding. And then, this summarizes our new information about where this P8 protein binds to its Lewis positive secretors. So here we're comparing, we have the P3 genotype, and it has some overlap with this site, but it's a little bit in a different location. Here we have P11, it binds in the beta-beta edge, but it really stretches out into a different part of the protein as well. Here we have P4 was a crystal structure determined by another group, and but this is with a Lewis negative LNFPI glycan, and it binds over in this beta alpha site. And then here is our new predicted location here of where P8 binds in the beta-beta edge to the Lewis positive glycan. So we were, again, fortunate enough to solve the crystal structure of the P8 VP8 star receptor binding domain with this longer glycan that contains both the secretor fucose and the Lewis fucose. So now we have the atomic resolution details of where this molecule binds, and we map out all of the specific interactions. And now we're able to really compare in great detail for P8's binding interactions. And one thing that's quite interesting is if we look at P8, it also binds to the Lewis negative glycan over here. Let's see. And then the Lewis positive glycan, it binds over here on the beta-beta site. So what we're seeing is it appears to be a theme is that each of these genotypes retain the capability to bind either Lewis negative receptors or Lewis positive receptors in a different binding site out of one of these two possible binding locations, either in the beta alpha binding site or the beta beta binding site. Now, the last thing that that I'm going to show here is that we did measure the KDs for all of these various combinations. And you'll notice here we have P19, and these are millimolar dissociation constants, so they're not that tight. It's kind of ideal for measuring by NMR, I suppose. But each one kind of has its own preferred glycan ligand. So we have P19, it has the tightest binding to a Lewis B negative and a secretor positive phenotype. P6 binds tightest to a double negative. And then we have P4 and P8, the ones that primarily are most responsible for human infections, binding most tightly to the Lewis positive and secretor positive glycans. So again, this kind of reinforces our idea that this evolutionary branch from P1 to P2 is evolving under the selective pressure of the more mature glycans that are observed in humans and moving from animals. These ones that are Lewis negative and secretor negative have the least mature glycan structure. And then as we begin to add the secretor fucose and the Lewis fucose, we consider those to be more evolutionary mature histoblood group antigens. So with that, I'll just go over our conclusions. We were able to report 2 critical crystal structures that detailed a new binding site for the P19 genotype, and then the first crystal structure of a P8 genotype bound to its glycan ligand. We reported both the STD NMR and the HSQC chemical shift perturbation studies for all 4 of the human genotype RVs. We will also determine the dissociation constants for glycan binding for, again, all 4 of the human genotype RVs. And we conclude that the evolutionary stage of the histoblood group antigens, i.e. the presence or absence of the secretor and Lewis B fucoses, determines the binding specificities for the different P2 genogroup rotaviruses. And we also determined that it appears that all the P2 human genotype or the P2 genotype rotaviruses appear to retain both a beta alpha and beta binding site for recognition of Lewis B positive and Lewis B negative glycans. And so we feel that this data supports our hypothesis that a vaccine that represented all 4 of the human genotype RV strains would be required to prepare a universally effective RV vaccine. And with that, I will conclude. This is a rather outdated acknowledgement slide, but it does contain the 2 people in one slide who are responsible for the work. So the first study with the P19 was done by Teresa Ramelow, who has since moved on. She was a postdoc at the time, and now she's at Rensselaer Polytechnic Institute. And then all of the remaining work was done by my postdoc Shen, who has done a fantastic job on all of this remaining study. And then our critical collaborator is Jason Jiang down at Cincinnati Children's Hospital Medical Center. And with that, I thank you and be happy to answer any questions.
Okay, thanks, Michael. Excellent talk. And I'm not seeing questions right now. If anybody has a question, please feel free to enter it into the Q&A box.
Can I ask a question?
Yes, Kathleen Hall has a question.
Yeah, so Michael, those affinities are very weak.
Yes.
Can you comment on the biological relevance? I mean, if you were to make a Yeah, they're very weak.
They are very weak. That's, yeah, definitely. The biological relevance of it, I'm not sure that I have a good explanation for why they're able to do what they need to do with such a weak binding constant. Certainly, we know that those are the essential receptors for infection, but why do they get away? It's possible that there is some work in concert with other factors, like the VP5 star domain. It may latch down and add to the affinity. It may not be solely determined by just the HBGA receptor domain. And I think there are also integrins that latch down onto the cell as well. So it may be that we're not seeing the entire picture by just looking at the VPA start domain.
Okay. Well, if there's no other questions, what I'd like to do is thank all three speakers in this session again. And just say myself, I've had a very enjoyable Saturday listening to some great science. And I think that's one of the exciting things about the Gateway Conference is that we get together once a year and share some exciting magnetic resonance spectroscopy. And from hearing all the talks today, I think I can conclude that magnetic resonance spectroscopy is alive and well in the Gateway region. And I think with that, I will turn it back over to Marcus, and I believe Marcus has some final comments.